Hello everyone. Today's topic is anemia. First of all, we discuss about anemia. What actually is the anemia? Anemia is a bleeding disorder characterized by reduction in RBC count, HP content, and PCV means packed cell volume. If these three contents are decreased in our body, we call it as anemia. Okay, how these three contents are decreased in our body due to the decreased production of RBC, increased destruction of RBC, and excess loss of RBC. Decreased production of RBC is mainly seen during any bone marrow diseases, and increased destruction of RBC seen in any diseased conditions or due to intake of any toxins that causes hemolysis of RBCs, and excess loss of RBC as seen in injuries or any of the accidents, or in such cases we can see the reduction in this abo contents. Okay, see, anemia can be either by inherited or by external environmental issues. Like inherited disorder is because of genetic variation or any mutations in the genes, and environmental issues such as nutritional problems, infection, or exposure to toxins or drugs. Okay, now, let us discuss about the classification of anemia. Anemia is classified based on the two classifications: morphological classification and etiological classification. Morphological classification. Morphology means what? External, like external appearance, no? External appearance, right? So, external appearance, we can see that RBC's size we can observe by externally, and the color also we can observe externally. What is RBC size? It is biconcave, right? And color is red. This red color is provided by HP. Yes. So based on this external appearance, which is known as morphological classification, it is divided into four types. In morphological classification, anemia is divided into four types: normocytic normochromic, macrocytic normochromic, macrocytic normochromic, and microcytic hypochromic. See, normocytic. Cytic means size. Okay. Chromic means color. Okay. See, normocytic, normochromic means normal size and the normal color. But in this anemia, there will be decreased production of RBCs. Okay. Remaining all are fine, like size and the color is fine. Next, macrocytic normochromic. Macro means large. The RBC will be larger in size, and normochromic means normal in color. Then macrocytic. Macro means same, larger in size. Hypo means less, no? So HP color is less due to the maybe the person has nutritional deficiency or any other cause. Means intake of iron, if it is decreased, then HB content will be decreased, right? So or any other conditions may leads to hypochromic condition that will be discussed later. Then microcytic hypochromic. Micro means the RBC size will be small, and the hypochromic means HP content is decreased. So the morphological classification we discuss it. Now etiological classification. Etiological means based on the study of cause or origin. It is divided into five types: hemorrhagic, hemolytic, nutritional deficiency, aplastic, and chronic. Chronic means due to the diseases for other organs or any other causes may cause anemia. Okay. So in detail, we will discuss separately about each of them. Okay. Hemorrhagic. Hemorrhagic means it is due to the loss of blood. Hemorrhagic means loss of blood. Loss of blood occurs for some time. It is called acute. If it occurs for a long time, it is called chronic. Anything that is last for a long time, it is called chronic. And if it for some time, it is called acute. Okay. See, uh, acute acute the hemorrhagic anemia has two causes: acute cause as well as chronic cause. Acute means sudden loss of large amount of blood, as in accident. In accidents or any injuries, there will be loss of blood, and then it will be replaced within some hours or some time, because of the clotting mechanism, right? Then, after the loss of blood, within 24 hours, plasma portion of the blood is replaced. Portion of the blood is replaced. Whereas RBC replacement take 4 to 6 weeks. However, morphologically, RBCs are normocytic and normochromic. Here, we have to understand that. See, the RBCs again formation takes 4 to 6 weeks, but the produced RBCs are healthy, means they will have normal size and normal color, so that the person has normocytic normochromic. But here we have to see that RBC take 4 to 6 weeks to reappear, right? So RBC content is decreased in that person, but the produced RBCs are healthy, like normocytic and normochromic. Then chronic means large amount of or some amount of blood loss for longer period of time. Okay. Then when there will be loss of blood, there will be loss of RBC. This stimulates the bone marrow to produce RBC, conditioned corrected within 4 to 6 weeks. This condition is corrected in 4 to 6 weeks. Loss of blood by internal or external bleeding over a longer period, as in peptic ulcer, purpura, hemophilia. Due to continuous loss of blood, loss of iron is lost from body, causing iron deficiency. See, in uh, chronic diseases like peptic ulcer and purpura, there will be loss of blood every day, like continuously there will be loss of blood for a longer period of time. So in this loss of blood will stimulate the bone marrow to produce RBC. But in this case, due to continuous loss of blood, there will be loss of iron. Okay. Loss of iron causes iron deficiency anemia, but the RBC production are not affected, because RBC is produced due to the stimulation of lost RBC stimulates the bone marrow to produce RBC. So RBC production is normal only, but the lost iron takes some time to replace. So the person will suffer from iron deficiency anemia. Okay. Next, we have to discuss about the hemolytic anemia. Hemolysis means what hemolysis means? Destruction of RBC. Okay, hemolysis means destruction of RBCs. Hemolytic anemia has two causes: extrinsic hemolytic causes and intrinsic factors. Extrinsic means other than RBC. No, there will be no problem within the RBC. Only the problem is hemolysis of RBC occur due to the external factors like antibiotics, drugs, any other. Whereas intrinsic hemolytic anemia means the problem within the RBC, means something mutations or genetic defects occurred in the DNA of the RBC. Okay, during the development. However, there will be no nucleus in RBC after maturation, but this is seen during the process of maturation. See, intrinsic hemolytic anemia. Sickle cell anemia. One of the example is sickle cell anemia and another example is thalassemia. Sickle cell anemia is a inherited disorder. In this, there will be abnormal HB. HB is abnormal, called as HBS. This abnormal HB is called HBS. In this, alpha chains are normal, but beta chains are abnormal. In this, alpha chains are normal, but beta chains are abnormal. So, the molecules of HBS polymerize into long chains and precipitate inside the cells. Because of this, attain sickle shape, become fragile hemolytic. See, actually, what happens in sickle cell anemia is there will be alpha chains are normal, but the beta chains are abnormal, and the molecules of HP are polymerized into long chains and precipitate inside the cells, which causes the RBC shape to get changed. It will become sickle in shaped. So, actually, the RBC shape is biconcave. Okay. So when there will be change in the shape of the RBC, the movement within the blood vessels become problematic. See, blood vessels in when they move in blood vessels, the sickle shaped RBCs get stuck in there, so that when they struck within the blood vessels that supply the bones or the organs get infraction, means there will be no blood supply to certain organs or certain bones where there will be the blockage by this fragile or this sickle shaped RBCs. Okay, so then there will be problem to the certain organs and certain bones where there will be this what aggregation of this sickle cells. See, in children, hemolyzed sickle cells aggregate and block blood vessels, leading to infraction. It is common in small bones. See, hemolyzed sickle cells aggregate and block blood vessels, leading to infraction, means stoppage of blood supply. It is common in small bones. See, in children, what happen if we take blood vessels? See, this is the blood vessels. See, RBCs normally biconcave, they will move easily. When we take, if this is blood vessel, these are fragile in shape, right? The movement become problematic, and the movement become problematic, and due to this sickle shape they cannot move within the blood vessels. So what they happen, they will get all of this sickle shaped blood vessels get sorry, sickle shaped RBCs will get aggregated within the blood vessel. This is a blood vessel. These are the fragile shaped RBCs. Sorry, these RBCs are sickle shaped, right? The sickle shaped RBCs are aggregated within the blood vessels. What happened? They become fragile, right? They get hemolyzed, means they get breakdown. Hemolyzed, this sickle shaped RBCs get hemolyzed. Hemolyzed RBCs aggregate and stop here only. Like they will block the blood vessels. Suppose this blood vessel, if you take this blood vessel, is supplying blood to the metacarpal bones. Okay, so when blockage is happen, then the blood supply is decreased to this metacarpal bones, so that metacarpal bones do not get nutrition. They get nutrition through blood vessels only. No, so now there is no movement within the blood vessels, so there will be stoppage of the blood supply to the that metacarpal bones, and there will be no nutrition, and the digits and the metacarpal bones where is their length, and the person will have this hand and foot syndrome. If it occurs for hand, it will be hand, and if it occurs for foot, they will have hand and foot syndrome. Okay, once again, the infracted small bones in hand and foot results in varying length of digits. This condition is called hand and foot syndrome. Okay, in children, hemolyzed sickle cells aggregate and block the blood vessels, leading to infraction. Means supply of blood supply to this metacarpal bones become stopped. Okay, stoppage of blood supply to these bones, so that there will be varying length of digits. This condition is called hand and foot syndrome. Okay, next, thalassemia. Thalassemia is two types, alpha and beta thalassemia. In alpha thalassemia, alpha chains are less, whereas while beta and gamma chains are more. In beta thalassemia, beta are less abnormal, while alpha are excess in excess chains will be seen. Okay, next, third division is the nutritional deficiency anemia. Nutrition means the food we take, right? When there will be decreased intake of particular nutrition to get matured of that RBC, if it is decreased, then we will call it as nutritional deficiency anemia. It is of some types. One of some of them I discussed. One is the iron deficiency anemia. See, iron deficiency anemia means in this the person has decreased iron content. We know that HB is made up of iron, right? So when there will be decreased content of iron, the HB will be decreased in that RBC. The person will have hypochromic conditions. Okay, hypochromic. The RBC will be decrease in color. Okay. What are the causes for iron deficiency anemia? Loss of blood, decreased intake of iron, poor absorption of iron from intestine, poor absorption of iron from intestine, and increased demand for iron conditions like growth and pregnancy. See, during loss of blood, normocytic normochromic condition. No, no, sorry. I discussed in this chronic condition of this hemorrhagic anemia. There will be iron deficiency anemia because loss of RBC is easily repressed, whereas iron is takes some time, so that the person has the iron deficiency anemia. So there is loss of blood and decreased intake of iron. Some persons food may not contain iron, that may cause iron deficiency anemia, and poor absorption of iron from intestine, and increased demand for iron in conditions like growth and pregnancy. During growth and pregnancy, we require more amount of iron. So in that, the person may have iron deficiency anemia. Then protein deficiency anemia. See, HB is made up of proteins, right? Deficiency of proteins that synthesize HB. HB formation, HB is synthesized from proteins, right? So when deficiency of proteins, then the synthesis of HB becomes abnormal. So in that condition, the person will have the protein deficiency anemia. The RBCs are macrocytic and hypochromic. Macro means larger in size. Hypochromic means there will be decreased color. Next, pernicious anemia. Pernicious anemia is caused due to the deficiency of vitamin B12. Vitamin B12 absorption occurs in mostly intestine. The intestine releases factor called intrinsic factor. Intrinsic factor is required for the absorption of vitamin B12. When there will be decreased production of intrinsic factor, we can see the pernicious anemia, or due to the gastric mucosa atrophy. When the mucosa becomes atrophy, then there will be decreased absorption of this vitamin B12, so that then there will be pernicious anemia. See, deficiency of vitamin B12, atrophy of gastric mucosa because of autoimmune destruction of parietal cells. Gastric atrophy results in decreased production of intrinsic factor. Intrinsic factor is required for the absorption of vitamin B12. When there is decreased intrinsic factor, then decreased absorption of vitamin B12. Poor absorption of vitamin B12, which is maturation factor of RBC. What is the purpose of taking vitamin B12 is vitamin B12 is helpful in maturation of RBC. Okay. Next, we will discuss about megaloblastic anemia. Megaloblastic anemia is due to the deficiency of folic acid. RBCs are not matured. DNA synthesis is also defective, so nucleus is immature. What happened? DNA synthesis become defective. So DNA is present within the nucleus, right? So nucleus also become immature. RBCs are megaloblastic and hypochromic, means larger in size. See, pernicious anemia appear in megaloblastic anemia also, because this absorption of vitamin B12 and this folic acids are correlated. That is called methyl folate trap. It will be discussed in the biochemistry vitamins chapter. That time you can understand the correlation between the pernicious anemia and this megaloblastic anemia. Next, aplastic anemia. Fourth type is the aplastic anemia. Aplastic anemia means in this, um, there will be bone marrow disorder. All of us know that RBCs are produced within the bone marrow. Bone marrow consists of red colored substance that produces RBCs. That bone marrow is reduced. Bone marrow is reduced and replaced by fatty tissues. The bone marrow consists of substance that produce RBC. That substance is replaced by the fatty tissues. So we can see the aplastic anemia. How this bone marrow is reduced and replaced by these fatty tissues, we will see in which conditions, how this what are the causes for this replacement of or reduced bone marrow, and we, how this how this bone marrow is reduced and replaced by fatty tissues, we will see. See, repeated exposure to the X-ray or gamma ray radiation. presence of bacterial toxins and quinine, gold salts, benzene, radium, etc. When the person undergoes frequent X-rays or gamma radiations have certain uh, characteristic which can damage this red bone marrow. Okay. Then presence of bacterial toxins and quinines and gold salts, benzene, radium, etc. This also cause decreased bone marrow. In tuberculosis, TB patients may also have aplastic anemia, and viral infections like hepatitis, HIV infections. These all conditions causes bone marrow to get reduced and replaced by the fatty tissues. In this aplastic anemia, the RBCs are normocytic and normochromic. In this, we have to understand that in aplastic anemia, bone marrow is reduced, so that there will be bone marrow is reduced, right? Then the RBC production is decreased, but the produced RBCs are normal. There is no problem in the RBCs. The problem is the production of the RBCs are produced in the bone marrow. The bone marrow has a problem. So there will be decreased production of RBCs, but the produced RBCs are healthy. They are normochromic and normocytic. But production of RBCs decreased. Okay. Next, fifth type we are going to discuss anemia of chronic disease. See, in this type, it is the second most common type of anemia seen in India. In this, in this case, we see that the disease of other organs or any other systems cause the anemia. It is secondary to these diseases. The common causes of anemia of chronic diseases: non-infectious inflammatory diseases such as rheumatoid arthritis. It is a chronic. This disease is discussed later in other chapters. This disease may cause the anemia too. Chronic infections like TB caused by Mycobacterium tuberculosis and abscess in lungs causes the anemia. Chronic renal failure. See, in we know that renal means kidney, right? Kidney consists of Jg cells. Jg cells produce erythropoietin. Erythropoietin is important for the stimulation of the bone marrow to produce RBC. So when the person has renal failure, then there will be no erythropoietin. Then there will be no stimulation for a bone marrow. Then there will be decreased production of RBC. See, in which erythropoietin secretion decreases. Erythropoietin is necessary for the stimulation of bone marrow to produce RBCs, and its deficiency causes anemia. And neoplastic disorders such as Hodgkin's disease and other diseases may cause anemia. So, till now we discussed that to classification, morphological classification and etiological classification. In morphology, four types we discussed, and etiological, five types we discussed. These are the different types of anemia. Okay, thank you.